So hello everyone and welcome to Fluid Sciences webinar. My name is Jenny. I'm currently the marketing executive at Fluidic Sciences, and today I'm very happy to moderate our session with the title Measuring Oncoprotein Copy Numbers and Antibody Affinities in a Native Lipid Environment. And today I'm very excited to introduce our keynote speakers, Professor Sandra Keller from University of Graz, Lena Bauernhofer from University of Graz, and our CEO of Fluidic Sciences, Dr. James Wilkinson. And before we start, let me run through the agenda. So James will be talking first, and he'll be giving you the introduction to the microfluidic diffusional sizing technology, a new technology for quantitative analysis in biologically complex native environments. And later on, Sandro and Lena will be following up and give you a deep dive into the MAFCON assay, which is for quantifying the membrane protein affinity and copy numbers. And they will also be sharing their work on extending the approach from 2D cell cultures to 3D organoid models. And we're gonna save the last 10 minutes for the Q&A session. So if you have any questions along the way, please send them in the Q&A box, which is right below your screen, because every attendees are on mute. So that's the best way that you can send questions to us, and we'll make sure that we'll go through them one by one during the Q&A session at the end. And if you want to directly chat with the speakers, you can also click on the raise hand button, which is also right below your screen, so that I will unmute you and let you directly speak with our presenters today. And without further ado, let's get started. I'll pass over to James.
Thank you, Jenny. Let me just get my slides on. So yes, it's a great pleasure to be here today. Normally I'm far too busy to do these things, but it's really exciting to be able to be here. I hate missing talks. So it's a great pleasure to be able to introduce Sandra and Lena shortly. But what I wanted to start to do is give a little bit of an introduction to our company, what we do, very basics on the technology, and then you'll hear much more interesting things from our colleagues later. So the title is Measuring Oncoprotein Copy Numbers and Antibody Affinities in a Native Lipid Environment. So this is sounds a little bit complicated. I'm really looking forward to knowing how you guys are dealing with that. But I think it highlights one of the neat things about our technology. We do have this ability to work in native environments. It's not limited to native environments, but one of our big attractions is the ability to do this. So a very brief introduction. I believe Sandra and Lane will be talking about their work done mostly on Fluidity 1M system. This is our product. I won't get too much into product details, but essentially it's an instrumentation that utilizes a technology called microfluidic diffusional sizing. This is a completely in solution technology and has an extremely high specificity, which is why we can work on these crude samples. Essentially what it does is it measures the size using diffusion, utilizing microfluidics. But with the molecular size, this is hydrodynamic radii, it allows us to then look at things like binding affinity, concentration, stoichiometry, and a lot of this more complex analysis, which will be introduced later, is really powered by these machine learning algorithms and our cloud support software. So very brief introduction to the company. My name is James. I joined the company back in September last year, and I'm a protein chemist by background, really. Worked for a number of different technology startup companies. And my passion is really taking very new technology that has a real exciting future, and working through early commercialization of that. So I've worked for a number of companies in that field. The company Fluidic Sciences, we are approximately a year old. I stole this LinkedIn post from David, one of our board members, the other day. And you can see somewhere on it, it says it's a 1-year-old post. So we celebrated our 1-year anniversary a few weeks back. And this company was formed to really commercialize this MDS, microfluidic diffusional sizing technology. It initially spun out of the University of Cambridge. What you see in the bottom is an extremely attractive chemistry department in Cambridge. There's a lot more pretty buildings in Cambridge, but it was fair to show the chemistry department. So this spun out of the group of Thomas Knowles many years ago. And now, as a company, Fluidic Sciences, we are based down in Royston, so towards the bottom left of this map. Cambridge, for those unfamiliar, is an extremely small city, but a really, really big biotech hub, and supports a wide range of companies and science parks around its circumference. So although we're based in Royston, we obviously like to claim we're in Cambridge, but we have this pedigree from the university. Now, where we fit in, and again, no apologies, I stole this diagram from the LMB website because it really highlights the challenges that a lot of us face. There are an awful lot of different parameters that we wish to measure when we're doing biophysical analysis. There are, fortunately, also some amazing technologies out there. But this diagram is simply to highlight that no one technology is going to solve all your problems. We will claim our advantages, as with every other technology, but really what we're about is adding to that toolkit, having that little niche where we can find some specific applications and support other things. But the thing to remember is you do need multiples of these methods. So where we fit in is on this left-hand side here. So MDS is looking at size measurements as described, but that gives us insights into aggregation. And then we typically use size to measure affinities, stoichiometries, etc. like that. The way we do this is using these proprietary microfluidics. What you see here is a simplified version of it, but essentially what we do is we pipette in a fluorescence probe, so a molecule that we fluorescently tagged, onto the top. That is then flowed into a flow buffer, which is coming in through the bottom. And the key thing here is we have laminar flow across that channel. We then measure the fluorescence at the end of this diffusion chamber at the top and the bottom. And because of the laminar flow, the only way of fluorescent molecules getting from the top to the bottom is through diffusion. So we measure the 2 points of fluorescence at the end. This fluorescence gives us the ratio and gives us the RH directly. If we see something then that is formed as a complex, it gets bigger, it diffuses less, we see more fluorescence on the top, and we can detect that increase in the RH. Now, what we do to determine the KD is we do this measurement then at a number of different concentrations of a binding partner. So we see the average size increasing from a baseline here at 3.8. It increases up to a complex size of about 6.1 nanometers. Key thing here is we get the KD, in this case 8.4 nanomolar, but we also get that size and the size of the complex. So this gives you confirmation, gives you confidence. It's that quality control that you've got the molecules you think you are. This is not aggregated because then it would be more than 3.8 nanometers. So you get all of these three parameters straight away. That whole thing takes about 25 minutes. And I'm just going to share this video. Hopefully this will come across over the webinar. But just to show you a little bit how you actually interact with the system. Most of it's done on the touch screen. You put your name of your labeled species, your concentrations, which buffer you might be using, all labeled on the touch screen. Sample preparation is the thing that then takes 5 to 10 minutes. We typically do a KD experiment by doing a serial dilution. We would then add in our labeled probe or labeled molecule, and then we load these microfluidic chips. So each of these chip plates you see takes 24 microfluidic circuits. We can therefore measure approximately 2 12-point KDs in about 25 minutes. Once that is finished, it all displays on the touch screen. You can get the results, you can manipulate like the sizes, things like this, but you can also look at the KD, the sizes all displayed in the top right. You can then also export that to your own machine via a CSV file. So the instrument I've briefly mentioned, the key to this technology is these microfluidic chips, 24 on a plate, very reproducible. It also means the instrument itself is fairly robust. No fluid really enters the instrument. This is a concept that will get mostly introduced later, but I wanted to briefly touch on it. And this is our cloud-based software. This is really utilizing our ability to measure and crude samples. Instead of doing a curve on the top left, for example, where we know the concentration of our binding partner, we do it in a way where we don't know it. Now, this curve then on the top left is essentially fairly meaningless. On the right-hand side, on the top right, you see all of the different combinations for concentration and KD that will satisfy this binding curve. So if we do not know the concentration of the binding partner, we do not know the KD. What we can do though is do another experiment at a much higher probe concentration, and we get something like in the bottom left. So now again, we do not know the concentration of the binding partner, but we've done the probe concentrations at 1 and 20 nanomolar. We see this curve shift significantly to the right. And now, using our Bayesian inference methods, we can determine a single solution for those two binding curves, which outputs the KD and, crucially, the concentration, and perhaps more crucially, the active concentration. So this is the generic assay, but you're going to hear a little bit more later about how this can be really put to sensible use. One example to finish before I pass on. This is the sort of data then you get from our system. What we're looking at here is a protein-protein interaction. We're looking at FKBP12, and we're increasing the concentration of FRB. What we see is that in the presence of 50 nanomolar rapamycin, we can generate a really nice binding curve with an affinity in the range of about 50 nanomolar here. Where we do not have the rapamycin present, even at very high concentrations of FRB shown in red here, we see a size that is just the FKB12 on its own. So we see no binding. So this is our ability to determine binding, KDs, yes or no, and see the influence of small molecule modulators on this process. And I think I will leave it there, because I'd like to pass on to the more interesting talk. So again, our key specs here, in terms of affinity range, we can determine affinities down to approximately 20 picomolar. We can go a little bit tighter than that as well, with a few tricks. Our system can distinguish sizes in the order of about 1 to 20 nanometers. So for those who work in proper numbers, that's equating to about 1.4 to about 14 megadaltons. 1.4 kilodaltons, sorry, up to 14 megadaltons. A single size measurement takes about 5 minutes, but if you want to do a KD experiment, perhaps 2 times 12 on that 24 plate, it takes about 25 minutes. And then all of this is collected with just 4 microliters of sample. So very quick method, very easy to use. We don't require any calibration to determine the size at all. This absolutely comes from the microfluidic fluorescence measurement, and our ability to measure size, affinities, stoichiometry concentration, or in cell lysate, cell serum, unpurified cell pools is a pretty powerful combination. So I think with that, what I'd love to do is to hand over to our guest speakers today. So Professor Sanjay Keller and Lena Baunhofer from the University of Graz. They've kindly agreed to talk about their work on oncoprotein copy number and using our system to look at antibody affinities in this native lipid environment. So thank you, first off, guys, for giving this webinar. Really look forward to hearing what you've got to say. Thank you.
Yes, thank you very much, James and Jenny, for the kind introduction. So Lena and I will be talking about how to measure oncoprotein copy numbers and antibody affinities in a native lipid environment, which we did, of course, using microfluidic diffusion and sizing. So in the first part, I give a brief introduction to the biological background, and Lena will take over and explain an MDS assay that we developed some 2 years ago, which we dubbed MAFCON. This stands for Membrane Protein Affinity and Copy Number. And Lena will explain how this can be used to study membrane proteins extracted from cell cultures grown in 2 dimensions. And then she also gives a brief outlook on how this can be extended in the future to 3D samples like tumor biopsies. And then I wrap up and briefly talk about a different MDS assay called full MDS, which stands for Fluorescent Universal Lipid Labeling for MDS. So the biological background, we were interested in a membrane protein called HER2. So HER2 is overexpressed in a number of cancers, including breast cancer, ovarian cancer, gastric cancer, and lung cancer. And HER2 belongs to the family of receptor tyrosine kinases. And there are 4 famous members of this receptor family called HER1, HER2, HER3, and HER4. And the curious thing about HER2 is that we do not know its endogenous ligand. It's very likely that it doesn't have an endogenous ligand. But nevertheless, in about 20 to 30% of all breast cancers, HER2 is overexpressed, and this overexpression is associated with poor clinical outcome. And HER2 has been targeted by the pharmaceutical industry for decades, and there are two major classes of drugs that bind to HER2. These are monoclonal antibodies on one hand, and tyrosine kinase inhibitors on the other. So monoclonal antibodies come by fancy names like trastuzumab and pertuzumab, and those antibodies bind to the extracellular domain of HER2 and thus inhibit or at least reduce cell signaling. The two antibodies that you can see, trastuzumab and pertuzumab, bind to different epitopes, so they have slightly different modes of action, which will be important later, because HER2 can undergo so-called ectodomain shedding, which means that the extracellular domain is cleaved off, and this means that the protein will no longer be recognized by pertuzumab, but it will still be recognized by trastuzumab. The second flavor of drugs that have been developed against HER2 is so-called tyrosine kinase inhibitors. So those are small molecules that bind to the intracellular kinase domain of HER2 and thus inhibit ATP-dependent phosphorylation. And the specific tyrosine kinase inhibitor that we studied is called lapatinib, and lapatinib binds to the inactive conformation of HER2. And this will be important later because it means that dimerization of HER2 will be prevented once lapatinib is bound. And this also leads to the disruption of signaling pathways and to the reduction of cell proliferation. Now, why would we need a new assay to look at HER2 copy numbers and HER2 antibody interaction affinities? I mean, currently there are two major methods that are used diagnostically. So if a patient is diagnosed with HER2, the first test that's done is immunohistochemistry. The problem with immunohistochemistry is that it's pretty crude. So there's a scoring system. The pathologist looks at the tissue sample, at the staining intensity, and then rates it as 0 if the staining looks like healthy, normal tissue, or +1, +2, +3, depending on the degree of staining. This is a very well-established qualitative assay, but it's not quantitative. The second assay that's established is quantitative. This is FISH, fluorescence in situ hybridization, but FISH quantifies gene copy numbers, not protein copy numbers. So this is usually done once a patient has been diagnosed to be HER2 positive using immunohistochemistry. The problem here is that a gene copy number might be relevant for protein copy number, but there is no one-to-one correlation. And this is why we set out to develop an assay that can determine HER2 protein copy numbers in tissues and in cell culture samples. And with this, I'd like to hand over to Lena, because she's the one who established everything and who's doing this experiment every day.
Thank you, Sandro. So I want to dive right in and tell you about the MAFCON assay. So MAFCON, as Sandro already said, stands for Membrane Protein Affinity and Copy Number. And I quickly introduce you to the assay design that we need for this assay. So we start with prepared membranes from cancer cell lines. And in our case, those cancer cell lines overexpress the protein of our interest, so HER2. And to those membranes, we then add polymer or some other nanodisk forming surfactants, and we end up with a native membrane protein library. So this nanodisk library does not only contain HER2, so the protein of our interest, but the whole membrane proteome of the cancer cell line. And what we do then is we titrate this nanodisk library to fluorescently labeled antibodies that selectively target HER2. And those complexes we then measure. So we measure the size of those complexes, so antibody to antigen in nanodisk with MDS. And this was already explained to you in detail in the first part. So we start right in with the results that we get from this. So here you can see a typical binding isotherm that depicts the binding of trastuzumab to HER2. And as you can see here, as the saturation of the antibody is achieved or approached, we get an increase in the hydrodynamic radius, because the size always reflects the average size of the population. And for trastuzumab binding to HER2, we get here a KD, so a dissociation constant of 0.5 nanomolar, which means that this is a really strong binding from trastuzumab to HER2. And we then, with the help of the MAFCON assay, gathered that there are approximately 20 nanomolar of HER2 in this cancer cell sample. And in the last few days or weeks, we also started to try clinical inhibitors. So you can see here again the measurement of just trastuzumab with the cancer cell lines that highly express HER2, where we have a KD of 0.5 nanomolar, 20 nanomolars of HER2, and this then translates to approximately 4 million binding sites per cell. So 4 million copies of trastuzumab can bind to HER2 in this specific cancer cell line per cell. And if you then add lapatinib, the tyrosine kinase inhibitor that binds to the intracellular domain, you get a similar KD, but you get twice the number of binding sites per cell, which could mean that as the inhibitor stabilizes the inactive form of HER2, and HER2 needs to be a dimer in order to be active. This could mean that it also disrupts those dimers in the nanodiscs and creates more binding sites per cell. Another interesting thing that we can measure with MDS and with the MAFCON assay is the measurement of full length versus truncated HER2. Sandro already explained this very briefly, but I will give you a short recap. The two antibodies that are currently most used for patients are trastuzumab and pertuzumab, and those two bind to different epitopes of HER2. So while trastuzumab binds near the transmembrane region, pertuzumab binds to the far end of the ectodomain. And in the case of ectodomain shedding, this domain gets shed off by enzymes, and with the help of MAFCON, we could measure that there is approximately 4 to 5-fold more truncated, so shed off HER2 than the full length HER2. And this means that there is 4 to 5-fold more HER2 that is still functional, but no longer approachable by those clinical antibodies. And now I will give you a short outlook on the next steps and what we are currently doing in the lab. So I explained to you those 2D model-based MAFCON experiments, but we are currently working on expanding this assay also to 3D models, because we want to go to a more native approach. So currently we're working on multicellular tumor spheroids, short MCTS, and we are trying to solubilize the spheroids without any further purification and create nanodiscs out of that. And the end goal would be to solubilize biopsy tissues from cancer patients directly, and then create nanodiscs out of this, and be able to, in order to be able to measure HER2 copy numbers in those samples. So the first steps are always solubilizing the tissues, or in our case, the spheroids, which we did here. And the first step is always to check if there are nanodiscs, which also can be tricky for biological samples, but Sandro will go into detail about this in the next few slides. So here we just did a size check of the nanodiscs with unspecific lipid stain and the MDS, and we can see here that approximately all of the cancer cell lines and the co-cultured cell lines that we had with spheroids, so you can see here, have a size of approximately 3 to 3.5 nanometers for the radius. So those are definitely still in the range of the size what we would expect for nanodiscs to be. And the next step after checking and making sure that there are really nanodiscs is measuring binding of trastuzumab to HER2. So here we just incubated those nanodiscs with trastuzumab and then measured the difference in size from just the nanodisc to nanodisc plus fluorescently labeled antibody. So we changed here what we labeled. And you can see a clear increase in size for overexpressing cancer cell lines of HER2, so those HCC1419 and SKBA3 cell lines have an overexpression in HER2, while we have here NHDF, which are just normal human dermal fibroblasts that have HER2 expressed but not overexpress HER2. So we would expect this increase. And the next steps would be to also measure those binding isotherms with the 3D culture nanodiscs. And now I will again hand over to Sandro, who will give you introduction to full MDS.
Yeah, thanks, Lena. So as Lena just explained, we often encounter situations where you want to determine nanoparticle, nanodisk sizes in a crude mixture like a cell extract. And this is why we developed an MDS-based technique called full MDS, which stands for fluorescent universal lipid labeling for microfluidic diffusion sizing. So the idea is that we replace more established techniques like dynamic light scattering, for example, or negative stain, electron microscopy, or cryogenic electron microscopy, because those techniques are really quite complicated or expensive, as is the case for electron microscopy, or they are susceptible to big aggregates, as is the case for DLS. So in our lab, we do a lot of dynamic light scattering, and this looks great as long as you have to deal with purified systems. And this is the example that you can see in the upper plot. That's a synthetic lipid in combination with a synthetic polymer. So those are chemically well-defined nanodiscs, and DLS is a great tool to look at such nanoparticles. The problem is that this no longer works for crude extracts from cells. So in the lower panel, you see nanodiscs that were made from a cancer cell line using a polymer called glycodipma. And we know from various other techniques that there are nanodiscs in this sample. We know this from electron microscopy, for example, from size exclusion chromatography, but dynamic light scattering is faring very badly with such samples because the size measurement is skewed towards large particle sizes, which dominate the signal. So this is why we developed full MDS 2 years ago. This is already published, and I just would like to focus on 2 exemplary data sets and briefly explain the assay. So full MDS is super simple. We again start with a membrane preparation or with cell culture extract. We then add a nanodisc forming polymer, a small molecule amplifier, to create nanodiscs. And then we add a fluorophore, such as Nile blue or Nile red, that petitions spontaneously into those nanodiscs. And the nice thing about Nile blue and Nile red is that those fluorophores are essentially dark as long as they're in the aqueous phase, but they become very bright, so the quantum yield increases by many orders of magnitude as soon as they petition into a lipid bilayer. And then we can use those non-specifically universally labeled nanodiscs to do microfluidic diffusion sizing. And on the next slide, you see an example of raw data on the left. So these are the diffused and undiffused channels in an MDS experiment. And on the right-hand side, you see the particle size in terms of the diameter that we got from MDS compared with the size distribution obtained from dynamic light scattering. And you see that in this case, so in the case of synthetic nanodiscs, the 2 techniques agree very well in terms of hydrodynamic size. But if you now turn to a more complex sample, or to 2 more complex samples, namely to nanodiscs made from E. coli, cells or from a melanoma cell line called WBN-004, then you see that in both cases, dynamic light scattering fails to pick up those nanodiscs, even though we know from lots of other techniques that the majority of the lipids and the proteins are found in nanodiscs. And this is in stark contrast with the MDS experiments. In both cases, you see that MDS yields the correct particle size of about 10 to 11 nanometers, and MDS is not affected at all by the presence of those few aggregates that screw up the dynamic light scattering experiments. So to conclude, what Lena and I tried to get across is that MAFCON, membrane protein affinity and copy number assays can measure the therapeutic antibody affinity and thus the cellular copy number of a membrane protein within a native lipid bilayer in so-called native nanodiscs. And with full MDS, fluorescent universal lipid labeling for microfluidic diffusion and sizing, we can get fast and straightforward measurements of nanoparticle sizes without the need to purify and covalently label those nanoparticles. So the next steps include screening other cell lines, other antibodies, including bispecific antibodies, for example, and also small molecule inhibitors, particularly RTK, so receptor tyrosine kinase inhibitors, and other types of membrane mimetic nanodiscs. And as Lena already explained, we will expand this assay from 2D to 3D systems to look at the HER2 levels and levels of other antigens in 3D cell culture models and then later in tissue biopsies. Okay, last but not least, I'd like to thank all members of our team who contributed to this project. Of course, our collaborators at Fluidic Sciences for the great collaboration over the past year, and our other collaborators who helped us with setting up cell lines and provided nanodisc forming polymers and small molecule amplifiers. Yeah, and thank you for your attention, and Lena and I look forward to taking your questions.
Yeah, thank you, Sandro. Thank you, Lena, and thank you, James, for your wonderful presentation. So now we move on to the Q&A session. I think we received a few questions in the Q&A box, although James has answered them, but I will read them out loud just for people who watch the recording. They will also hear about the questions. So the questions we have, first question we have is that, can this be done with primary cells or tissue biopsies, or is it limited to cell lines? I think for this question.
Yeah, so I've answered a couple on text. Yeah, fundamentally, we don't mind. The cells have to be lysed. We have to clarify them, spin down all of the insoluble bits. But then from our perspective, wherever the cell comes from, we don't mind. Not my field, but the advantage, of course, of using cell lines is that they can be a little bit more reproducible, experiment to experiment, perhaps. However, if you want to look at patient samples, because that's what you're really trying to do, and this is the ultimate goal behind a lot of this research, of course, then that's more than doable. We already work with patient samples using serum, blood samples, biopsies. We've done some cerebrospinal fluid work with patient samples. So yeah, none of that is a problem.
Yeah.
And I think also just one last point, the organization, if you like, of the system, because all of the sample stays on these microfluidics chips, which are disposable, then it makes potential contamination issues a lot simpler to deal with. All the sample just gets pipetted in that chip. You don't need to worry about cleaning between patients or anything like that.
Okay. Thank you, James. The next question is there a plan to support other membrane environments beyond glycodipma, for example, SMA-LPs?
Again, yes, I would say we already support it. I haven't come across anything yet that interferes with both our fluorescence channels where we use 488 and 647. Very high backgrounds. How to put this? So yeah, our spec on our instrument, we can detect 100 picomolar of Alexa 647. So this is really, really minute amounts. If you're working in full cell lysate background, you might need to use a bit higher than that, but we're talking single-digit nanomolar. So yeah, any of these more complicated polymers, but also traditional detergent molecules are all fine.
Yeah.
If I may add to this, We actually have tons of data from other polymer nanodiscs and also from small molecule amplifiers. And all of these compounds are compatible with MDS and MAFCON specifically. So here in this talk, we just focus on one polymer, which is glycodipmer, but it works equally well with electroneutral polymers, with DDM, the desmaldoside, as a conventional detergent. So the technology has no limits as far as the membrane mimetic is concerned.
Great. Thank you, Central. So next question is, can MAFCON be applied to GPCRs or ion channels?
Yeah, very simple answer, yes. Yeah, I mean, it can be applied. I mean, that assay, or the basis of that assay, can actually be applied to anything. It doesn't need to be a membrane-associated protein, technically. Now, the way we've set it up for the membrane proteins, these are the numbers you put on, and this is what gives you the copy number, as was so eloquently shown. But you could actually use it for any protein, really.
Okay, thank you. So the next question is, how does embedding proteins in glycodipmer nanodiscs preserve native conformation and function?
I will defer to experts on that question. I'm aware they do, but I'm not a membrane protein biologist.
I think this one is for Lena. She does it every day, multiple times.
So basically what you have with a nanodisc is you don't extract the membrane protein out of the native bilayer, but you essentially, with the help of the polymer, you cut out little pieces. You can imagine it like a little cookie cutter that goes to the membrane, then cuts out the membrane proteins, but also a part of the bilayer, and then the polymer wraps around this little piece of membrane like a belt. So the protein is still in its native environment, and it's just accessible to both sides, so the intracellular as well as the extracellular part of the membrane. So and there can be one protein in a nanodisc, there can be no proteins, there can just be like membrane, but there can also be protein complexes in this in those nanodiscs. So I hope that answers your question.
If I can have a follow-up to that, actually. So I mean, as a simple biologist, I work on proteins that are all very fixed and very, one or two proteins is how I tend to think. But there must be thousands of different lipids in varying lengths and bits and pieces. Are there 2 or 3 key lipids for the structure and function of a membrane protein, or is it a little bit more subtle than that? Do you need hundreds of different ones? I know basically where I'm coming at from this is it easier to extract proteins with their native lipids, or could you add in afterwards the 1 or 2 that you thought were critical that you could buy from Sigma and add them back in?
Yes, I mean, if you know that your pet protein depends on certain lipids, you could add those lipids. You can get them from a commercial supplier and add them in later. Or even better, I mean, that's what we at least hope, is that the protein would pull those lipids with it into the nanodiscs. I mean, this is a very protein-dependent question, right? With some membrane proteins that are very sensitive and require specific lipids. Other membrane proteins seem to be more tolerant in terms of lipid preferences. But the idea here is that since we extract those proteins from the native cellular environment, at least in principle, they have the possibility of co-extracting the lipids that they require. And this seems to be working very well, at least in the case of HER2. We saw that, I mean, from this comparison of binding isotherms with and without lapatinib, it seems that within those nanodiscs, in the absence of the inhibitor, HER2 is predominantly homodimeric. So extraction doesn't abolish those dimers. The protein remains homodimeric, and it is only upon addition of the inhibitor that the dimers dissociate. Right? And this is very different in harsher membrane mimetics like detergent micelles, for instance, which either fail to completely extract the receptor or which lead to receptor dissociation or even inhibit the receptor antibody interaction.
Yeah. Thank you.
Okay, thank you. So the next question we have is, can you use MDS for small molecule protein interactions determination? What is the size limit for the small molecule? Yes, this for James.
Yeah, that's a good question. So two answers to the question, just to make sure I've got it right. So the smallest thing we can actually see and determine the size of is just less than 1 nanometer. So if you have a fluorescent molecule that is about 0.9 nanometers, we can detect that and tell you it's about 0.9 nanometers. Now, if we have a fluorescent molecule that is 0.2 nanometers, what we can do is we can detect the fluorescence and tell you that it is smaller than 0.9 nanometers. So it will still work on the system, it will still go through, we will still detect it, but we will tell you that it is smaller than 0.9, and we can't have any higher resolution than that. Now, if that 0.2 nanometer particle then binds to a membrane protein in a nanodisk, the overall complex size will of course increase, and then we will see that increase. So you can still work with it, but you can't size it that small. The other way around then is the size difference in order for us to get a good signal and see. For that, we need a size change in the order of about 10 to 15%. And to be clear, that is a size change, not a mass change. So if I go to the one extreme that maybe your question is touching on, if we have a 50 kilodalton kinase and we're throwing lots of small molecules at it in typical drug discovery fashion, we will not see a small molecule, 300 Dalton molecule bind to a 50 kilodalton target. We will, however, see the result of that 300 Dalton molecule if it causes the protein to dimerize, or if it forms a complex with another heterodimer, or if it causes a homodimer to fall apart, for example. So we can see the effects on it. But yeah, size change, we need about 10 to 15%.
Okay, thank you, James. So for the final question we have is, how long does the entire workflow take from sample preparation to result?
Again, I think that's one for Lena.
Yeah, so it takes me like 2 days, but it's not the whole day. So I start with, I mean, if you have the cells ready, cell culture is always tricky, and I don't want to give any times on that, depending on how good your cells grow. But if you have your cells ready, then I take 1 afternoon for the preparation. I incubate the solubilization overnight. So I incubate the polymer overnight, and then I finish it off. So, and then you can start labeling. So I'd say 2 days approximately. And by the end of the second day, you can have already MDS data. So.
Fantastic. And again, to be clear, that's the sample prep, that's the equilibrating the samples, letting them all bind, and then the instrument itself, then you're talking 2 or 3 plates, something like that.
Yeah.
So a couple of hours running on the instrument.
Yeah. So that will be from 1 day to, from cell culture to data on the MDS is 2 days.
Amazing.
Okay, great. So I think we have answered all the questions we have for a Q&A session today. So it was very engaging Q&A session, very amazing presentations by everyone. And I would like to thank you for spending your time with us. Oh, I see there's a final question before we end. Yeah, let's try to address this. So can we determine the A, beta, or other fibrosizings or oligomeric states?
Yeah, I'll take this one. Yeah, someone's obviously been at our website, which is very, very good to see. Yes, we can. So it uses a very similar cloud-based or Bayesian inference approach that that Lena was talking about, but it does it in a slightly different way. So instead of the unknown concentration, as we had in this example, what we do is we take the known concentration, but then we simply compare the known concentration that we've measured with the active number of binding sites that we determine, and that's what gives us that stoichiometry. The oligomeric states then come from the size. So the size we can measure fundamentally just as it is. We do get an average size to note, and of course, I think part of the problem, or part of the taxing research in that field is you get a very, very large heterogeneity in the fibril sizes you get. So we see an average size. But it doesn't mean we can compare samples showing one is on average larger than the other. Our resolution is not going to be good enough to see, is it 7 or 8? But our resolution will be good enough to show that this one is on average about 8, whereas the sample before was on average about 6.
All right, thank you, James. Is there any other questions from the attendees? Okay, so if not, let's wrap up here. I would like to thank the speakers. Thanks Sandro, James, and Lena for giving amazing talks today. And thanks everyone for joining us for this webinar. And we hope to see you in our upcoming session. Okay, so goodbye and thank you.
Thanks, guys. Bye.
Thank you.
Thanks,
Goodbye.
everyone. Bye.
Thank you. Bye.